So what we've been able to do thus far is we have 4 types of cord blood or umbilical cord stem cells, 2 types of placenta stem cells, 3 types of mesenchymal stem cells from bone marrow, fat, and dental pulp. We've also developed a clinical grade IPS, and I think this is a type of pluripotent stem cell that will replace the embryonic stem cell. We've also built cancer cell technology and cancer stem cell technology. We have immunological cell technology, and we have processes to differentiate these stem cells into specialized cells and tissues. And we have ability to expand these human cells. So what is the rationale for regenerative medicine, and what are the alternatives to an embryonic stem cell? So our current medical treatments are designed to ameliorate symptoms and prevent disease. But regenerative medicine, if you can partially or completely regenerate an organ disease, this will substantially decrease the cost. But many of those who are in the embryonic stem cell field are advocating to use an embryonic stem cell because their argument is that it can do everything. If you can do everything, then you have the ability to offer solutions to a variety of different diseases. But in 2007, Dr. Yamanaka from Kyoto, Japan, was awarded the Nobel Prize, subsequently in 2012, for developing what's called the IPS cell. And what the IPS cell does is basically takes an adult cell, typically a skin cell, and basically introduces a number of genes that reprograms that cell, that adult cell, back into a primitive embryonic-like cell. So it has all the similar characteristics of an embryonic stem cell, but it doesn't have the controversy because you're not having to use an embryo and having to destroy it. The problem with the IPS cell, when it was originally introduced, is that it has certain technical issues that have to be overcome. One, you use the virus to introduce those genes in, and so you can't use a virus because of the infectious complications. And the other is that, like an embryonic stem cell, when you put that IPS cell into a mouse, it also forms a tumor. So you have a tumor genicity issue that has to be solved. So in 2006, we got started on all our stem cell technologies, and we started, like every organization, with very humble beginnings. We worked with the local Catholic hospital at Mercy in Iowa City and many rural patients in Iowa. So we have created the largest adult stem cell, cancer cell, and immunological cell repository in the world. And many people are not aware of that. So, put this in context, it would have taken government and pharmaceutical industry over 10 years and over $100 million to develop that program. In fact, NIH tried to do that by creating a regenerative medicine center and putting $100 million into that. But more recently, over a year ago, that program has fallen apart, and the director of that program has left because of the problems of getting that program off the ground. Now, in contrast, it took us 7 years and less than $2 million to do this. So basically, it's more than just about money. It's about execution, and it's about doing things that are free of red tape and the bureaucracy that you see in large organizations and in government. So what we have done is created a clinical grade IPS cell. This cell is free of virus free. It also, we also do not use a gene that was originally used by Dr. Yamanaka called c-Myc. c-Myc is an oncogene which forms cancer. So we've been able to, and c-Myc was originally used because it was necessary in order to reprogram those cells efficiently into IPS cells. Well, we have developed a strategy in which we don't even need c-Myc anymore. Also, what was originally used were what we call feeder cells. These are animal cells that basically the IPS cells are grown on a lawn of these animal mouse cells to provide nutrients to keep these cells alive. Our system is feeder free. So that's important because you can't be developing a therapy that have contaminated mice in your product. We also done it in a way that has a lower manufacturing cost. And when you inject these cells into a mouse, no tumors. So we have taken care of a lot of the issues to create clinically grade iPS cells.